small inconvenience, okay, because I have some issues to extract this fiber. So the fiber that we extract was the uncompleted one, so I have to do it like this. Okay, like
First of all, everyone appreciate him. He came voluntarily to take today, unlike that. Yes.
as you can see, the pathophysiology and the mechanism of intravascular hemolysis. I don't see the need of explaining what hemolysis is. As you all know, it is the premature breakdown of RBC before it is supposed to be. It means before 120 days. So in our body, there are two hemolysis happening. That is intra and extravascular hemolysis. So we'll look into intravascular hemolysis first, and we'll go on to extravascular hemolysis. Okay, first of all, this intravascular hemolysis. As you can see about intravascular hemolysis is the process where red blood cells RBCs are destroyed inside the circulatory system. It is the blood vessels. As you can see here in the picture, red blood cells are destroyed and free HB and bilirubin or hemat are all directly released into the plasma. Okay, and as you can see in this picture, also we can see how like the lab diagnosis. Okay, as a part of the lab diagnosis, part of the diagnosis is here. As you can see, free HB will directly go to the plasma. That means HB will increase, and then another thing is haptoglobulins. Okay, I haven't write it here, but haptoglobulins are a protein, okay, that can bind free HB from the blood vessels, okay. So that will also, that concentration will also increase, and then bilirubin also will be increasing, and LDH also will increase, as LDH are also present in the blood cells. Okay. And these haptoglobulins, as you can see, it binds the free HB and it helps to replicate the damage. Okay. And this is the primary triggers of destruction. Okay. First of all, this mechanical shearing, like small blood vessels, okay, MHA, microangiopathic, hemolytic anemia, like EIC or TPP, that means thrombocytic, thrombocytopathic purpura, okay, or biomechanical heart valves. And then the next one is complement activation. Complement activations are that MSC, membrane attack complex, which includes all that C1, C2, C3 till C9, okay, that forms on the RBC surface and hunting hole in the membranes and making them fragile and lose their cell integrity, which cause the cell to burst, okay, easily to burst. And then osmotic lysis is diffusion of solutions. It means the cells will swell and it will eventually explode and burst, okay. And direct toxins like Clostridium difficile or autoimmune reactions also like immune compatibility can also trigger intravascular hemolysis, okay. And then these are the mechanism of intravascular hemolysis, and here you can see it flow, okay, by mechanical shearing, complement activation, and coupling. And the membrane, okay, it loses their cell integrity, which makes them prone to, how would I say, it makes them prone to burst cases of some complementary activation will also attach to the red blood cells and which eventually will lyse the red blood cells, okay. And then I also put some small points about their biochemical consequences, okay, in intravascular hemolysis. That means hemoglobinemia and haptoglobin depletion. As you all know, HB will increase and haptoglobin will also decrease. And then hemoglobinuria, hemosiderin will also increase. And enzyme releases like LDH, okay, LDH is, as you all know, it is present inside the RBC and it will also increase. And unconjugated hyperbilirubinemia, as you all know, red blood cells break down into two parts, okay. And this is the lab diagnosis of intravascular hemolysis, and these are the EBS, okay, peripheral blood smear or microscopic examination. Presence of histiocytes in the hole marker, okay, and positive hemosideruria, that means urine in blood, okay. And then reticulocytosis, that means RE cells, reticulocytes also increase in intravascular hemolysis. Because inside RBC, inside the blood vessels, RBC breakdowns occur. So in that, the blood vessels, the blood loss to be compensated by the bone marrow. So increased RD cells will also be there, and more biochemical markers are serum haptoglobin will decrease, and elevated LDH and bilirubin that is unconjugated, which eventually will cause jaundice, and then hemoglobinemia and hemoglobinuria will also be there. Okay, and then like some mechanical trauma examples are, as you know, DIC, disseminated intravascular coagulopathy, and related are VNS and it's called autoimmune hemolytic anemia. And then severe infection like pseudomonas and because sorry, and then we'll go to extravascular hemolysis.
Okay. See, I'm not interrupting. Can you please tell me the concept of haptoglobin? We are all learning together. I'm not challenging anyone's knowledge. Tell me, what is haptoglobin? Why it is decreasing? What you are trying to say there?
As you can see, when RBCs break down, say there is free HB, it means haptoglobulins are produced by the liver. So that free HB will bind by the haptoglobulin to prevent oxidative damage.
Okay. Whenever HB released, yes, that oxidative damage will, nullified by haptoglobulin released by liver.
Yes.
But you say haptoglobulin levels will decrease.
Yes. That means the free HB and haptoglobulins will form.
Oh, since they fused here, yes, their levels will be reduced.
Yes.
Correct. Very good. Good.
And extravascular hemolysis. As you know, extravascular is extravascular. It means outside the blood vessels. Examples are MPO, molecular thrombocytic system. Also known as reticular system. Okay, it means in liver. The principal organs involved in this are the liver, and then the spleen followed by the liver, and then the bone marrow. As you can see here, after the small pictures where all the damaged RBCs are phagocytosis by macrophages, which will eventually lead to RBC breakdown. Hemoglobin is the release of is the byproduct, is the product of RBC breakdown and hemoglobin and the honor, and then okay, we'll eventually move.
Okay, nice diagram. Go back, very nice. See here they are showing recycling of molecules also. Once released, it's released amino acids which are returned to circulation for protein usage. Iron again bind with ferritin, recycled in bone marrow. Only bilirubin, that is unconjugated, will be converted to conjugated and excreted. Very nice. You can use this in the exam also. This picture. Very nice.
And these are the pathophysiology and the primary key for the absorption of the triggers. And as you know, opsonization antibodies, typically IgG, it means called the blood cell membrane, which makes a cell delicious and highly prone to specific macrophages. Okay. As you know, macrophages are used to do phagocytosis, it means they kill or they engulf the cell. And then they do from the red blood cells, but they flexible to pass through narrow split like. As you know, red blood cells are a nuclear damage that don't have nucleus, so that they can squeeze through the small blood vessels or the wrist. So, with abnormal cells, they can't pass through the small blood vessels anymore. So, it will eventually lead to lysis of the red blood cells. Okay, and then intravascular inclusions like Higgs bodies. Okay, on, and then these are the mechanism of extravascular hemolysis. Almost same as the first. Yes, first. Actually, that red blood cells are sensitized by the IgG that will engulf by macrophages, in liver, bone marrow, and break down products. Yeah.
So mainly they are destroyed here, not inside blood vessel, but by the macrophages, and these macrophages migrated to spleen, liver, and bone marrow. That's why it's called extravascular.
Yes.
Very good.
And these are the diagnosis of the extravascular hemolysis. Okay. And as you can see, evidence increase RBC will increase bilirubin, then eventually to jaundice, and LDH also will increase. And haptoglobin increase, and BBS, lymphocytes, mutated RBCs, polychromasia, and then yes, say Cook test. Yes, Cook test.
So yes.
Cook test is the, how would I say, it is the
Autoantibodies targeted, sensitized RBCs.
this
So this is how we can differentiate intravascular from extravascular. Usually extravascular happen due to macrophage phagocytosis. That phagocytosis is due to RBCs are pre-coated with some antibodies. So yes, Coombs test will determine extra from intra. Coombs plays vital role because here antibodies coated. That's why this macrophage engulfed. So very good. Coombs plays a vital role here to distinguish. Good.
Yes, this is the overall. Okay,
Very good.
This hemolytic anemia framework is intra, extra, or acquired or inherited. Okay. This is particular smear Dennis form. If you look into the PBS, you will see this type of cells in hemolysis. And then these are the. Yeah.
So very good. In this now we are saying first of all what is the reason for hemolysis? There we are saying it's due to membrane effect, enzyme problem, HB problem, or due to these three are intra problems. Intravascular problems. Coming to the extravascular, extra problems can be due to antibodies, abnormal antibodies attacking by pathogens or due to some drugs, chemicals that lead to extravascular issues. Then here you can see extravascular causes due to these three issues, either liver, spleen or macrophages. Bone marrow can also be affected. Then intra, obviously blood vessel. Then compensatory, okay, this is how anemia will be compensated by bone marrow. Then he also given very good picture of the biochemistry smears, any RBC destroyed, okay, it can be schistocytes, scarjets, sickle or spirocytes, indicate hemolysis. Then to differentiate extra from intra, he is doing Coombs test. Well done. So you used the notebook LM, very nice. Even I liked it. Well done. Everyone appreciates here. Such a nice presentation. You made it better than PGs, right? This is how to the point. Good job.